Yeah, so we're live. We were talking about exposomics, yeah.
It's not that new. It's now becoming more and more sought after in terms of explaining and understanding gene environment interaction, looking at chronic diseases, understanding what people are being exposed to. It's not easy. I mean, if you think about the genome, which is, from an individual is finite, right? But the exposome is infinite. Everybody is being exposed. I mean, just think of the temporalness of what you're exposed to every day, right? And so, how to capture that, how to quantify that. Um, so it's a pretty, but to understand disease, it almost to understand if you think of disease or some pathology, any kind of phenotype, to have the genetic component and then also the environment component, you know, it completes the understanding of what is driving
Mhm.
that particular disease. Yeah, so super essential. Right now, they're looking to start the human exposome project to complement the genome project. Yeah,
But when you say exposome, it's like what type of molecules are you talking about?
when I talk about exposome,
Chemicals, what could be like chemicals like molecules from naturally like occurring molecules? What is it?
it could be everything from viruses to the biological exposome, mycotoxins from fungus itself, um, virus, parasites, to it could be the kind of physical exposome, which kind of uh, chemicals are in that domain. Chemicals from food, chemicals from environmental pollution, right? The pollutome, plastics. So you can think about it like chemicals. We have a diverse set of chemicals, right? Everything from, like I was mentioning, like food, solvents, heavy metals from if you work in a car factory, or if you're by a volcano, or if you eat a lot of chocolate, right? You're gonna have some like heavy metals, and there's in during that exposure, right? Um, and so my group focuses on chemicals. It's probably the most studied because it's the easiest to study. But there's a lot of groups looking at even the social environment, right? Behavior, people who take drugs. If you think about it, even though we have chemicals in the physical, like the way we take prescription drugs versus what we're exposed to are two separate, different domains, because taking drugs, illegal or prescribed, that's a behavioral, right? Behavioral domain, or element to that exposure. So you got to think about that as well, right? So that's kind of complex, but more people are exploring different environmental domains, right? So when I say environment, we can quantify the genetics, right? The heritability. But then when we talk about the environment, of G by E, there is an endless amount of environments, right? And so we use environment kind of broadly. And for us, when we look at for example, we're going to look at we have the genetics of the individual, but we're going to look at the pollutome, all the pollution, all those exogenous chemicals. Then it's going to be gene by pollutome, interactions, right? So we define it better. Yeah. So that'd be and so that's one thing to like think about. You know, but I yeah, that's important, but we have to also, I think, the social determinants of health, right? Stress that comes from, whether it's warfare, right? The people, that exposure, right? That's probably also chemical, and stress in a different domain, right? Yeah, so it's really complex. It's really complex, but there are these mathematical models, statistical models that can try to capture that environmental effect, right? And that's just going to help us better understand disease, and health. Yeah.
So yeah, even stress is considered part of the exposomics, so it doesn't have to be endogenous, like exogenous molecules.
Yeah, yeah,
Okay.
yeah. Well, I mean, if you think about it, like there is an exposure or stress of some sort, or whatever that exposure can be internalized, and then you'll have, like, for example, higher cortisol or inflammation, right? That's a stressor on the body. So there's this kind of like, you know, niche selection, adaptation, and then something's happening biologically downstream. Now, and not everybody's genome is the same, so not everybody's response to that environmental stress is going to be the same, and all of those, like, you know, those features will have massive amounts of variation, right? But it's really, if you think about a simple model, right? Exposure, biological response, outcome, right?
Okay.
So that's good. Yeah. So yeah.
Yeah. So it's simple, but it's complex at the same time because you have, as you said, I mean, if you factor in stress and pollutants and biologically like normally occurring things, like for example, something that can come from a mushroom or a yeast, all that is considered part of exposomics, from what I understand, and that's a huge set of molecules.
Yeah. Right. Yeah. Um, yeah, no. So like, what's the contributing factor, right? What is the, how do you, if you, let's say you captured an individual, or let's say, let's say you were able to really capture throughout your lifespan the exposure, right? The most perfect experiment. At what point are those exposures based on your genome, right, being accounted for maybe something that you're experiencing, right? Whether maybe something that led to depression or something that led to you being sick, right? Um, the question is, uh, what is the link of those exposures to that metabolic response and the outcome, right? And obviously, there's going to be a lot of noise in there. So, and think about it, everybody will have like a genome that will be different in response to some type of exposure, right? And I think it's only a matter of time using AI, which is mass spectrometry is a pillar of the exposome, but AI and data science is right there. It is, yeah, imperative now, because there's so much data. There's going to be so much need to understand and see what that is. And then at some point, what is, how do we extract meaning? And knowing the genome of the individual, but knowing the exposome, my area, well, something I'm interested in, working with others, is how can we come up with an exposomic score, and how can we maybe rank or prioritize what are the more severe exposures, right? I can imagine in the future where somebody has, they have their DNA sequence, and then they're already doing exposome-wide studies, right? To look at this, what are the contributing, what are the things you should avoid, right? Maybe you're more sensitive to heavy metals, maybe you're more sensitive to like other types of pollutants, but maybe not. Maybe you're super resilient, but maybe a mushroom will take you out.
Hey everyone, this is not an ad. Go to blueword.com/in, that's BLUWR.com/in, like inside, and write about something that makes you think.
Basically, what we were saying is exposomic is the study of everything to which you might be exposed. I mean, where you are exposed towards your life. So that's pollutants like chemicals. It could be molecules from yeast. Could be like also the stress, like cortisol, like emotional exposure, all of that. But um, Oh, let's talk more about the chemicals. Like
Okay.
we can talk about the cortisol and stuff like another time, because I think what you're doing. I mean, I might be wrong, but what you're doing is more the study of the molecules.
Yes.
Yes. Okay.
Yeah.
So and you're a mass spec specialist.
Yes.
So mass spec is mass spectrometry. So we have to we also have to explain that. So if you can like very quickly explain what mass spectrometry is.
Yeah. So mass spectrometry is basically a technique where we can basically go from the either liquid phase or gas phase to and then ionize these molecules and then analyze those in a mass analyzer and detect them. Every molecule has a mass to charge, and it allows us for that capability. That's pretty much in a nutshell what a mass spectrometer is it allows us to get a reading. There's other components there like retention time, but the mass to charge will then be, once you have the mass, then you can have an idea of what the identification of this compound is. For peptides, it's a little bit more straightforward. It's easier for sequencing, but that's pretty much how. And you can mass analyze small molecules, peptides, proteins, larger complexes. But yeah, so it gives us that ability for like molecule identification. Yeah.
Okay. Very cool. So the aspect I think what you're really interested in when it comes to exposomic is the interaction between the genome and the exposome.
Right.
And because as you said, the genome is finite. I mean, in the sense that it doesn't change during your life. It doesn't change significantly. I mean, you must still get some mutations and that you don't want. But anyway, but it doesn't really change significantly throughout your life. But the exposome might, and
Well,
you have.
yeah.
It will, yeah, it will, definitely. It will change during your life. And so the idea is when you have a disease is to explain the disease not only on the basics, on the basis of the genome, it's like what genes you have and what mutations you have, but also how these genes define how you react to different molecules in your environment.
You got it.
So, yeah, okay. So yeah, that's fascinating. Because it means that it's not just your genome. Because I mean, the one of the major dogma was let's identify the genes, let's identify the SNPs. Like you have a disease, like SNPs is single nucleotide polymorphism. So let's identify all the SNPs, and then we can figure out the disease. But what you're saying is that it's not just that. That's not the whole story. There's another aspect. It's like what you've been exposed to during your life, and that might explain, for example, obesity. Like how likely you are to develop this type of problems. Diabetes also it might be one of them. So yeah. So how do you go about that during your research? So what's the plan?
Yeah. So, I mean, first of all, you can actually just look at the rates of diabetes, obesity, and they can't be explained by genetics alone, right? Heritability could already be calculated. So they're moving beyond normal rates of just genetics, right? So we know that there is an environmental response, right? You go about it by basically, so for example, developing basically a population study. Now that could come in a lot of different forms. Where we're at, we do a family study. We see a lot of related individuals, but you could do case control. You can do others kind of family designs. That's not my area. However, every kind of study design has its own kind of advantages, disadvantages. The advantage of a family study is the ability to see rare variants, more statistical power, right? So you can see, because go look at all of these, like, these big studies coming out of like the All of Us, and they need like several 100,000 participants to see a significant genetic effect or something, right? With us, we don't need that many. We have more statistical power with smaller amount of participants and subjects to sample or sequence, right? So we can look
because of the family study, because
exactly
the study is different. Yeah, but the family is not like 3 people. It's more like a few thousand people.
Yes, correct. Yeah. And you can account for relatedness, right? Yeah. And that's a whole nother area of research in itself. And I'm skipping out a lot of details, but what we do, our approach is number one, when participants come in, we phenotype them all, we take their blood, we can measure, for example, we can measure blood pressure, BMI. We can get all of these kind of physical characteristics of all of these individuals. We can measure liver stiffness. We could do MRI, right? And so we have all this phenotypic data, and think about, or we could take all these measurements, and some of these measurements may also even be readouts for diabetes, Alzheimer's, right? We can look at biomarkers. So once we have all that, then we can kind of see, okay, well, we have maybe 60% of the population has Alzheimer's, right? Obviously, if you adjust for age and all of these variables, right? But you can see like, oh, we have 50% are diabetic and also are obese, right? So also they have complete genome sequencing, right? So then you can go in and start to look at those same participants, the ones that have, or fatty liver disease is another one we look at, right? We look at liver stiffness, and we can look at fibrosis, and we can make these measurements, right? It's all about quantification of these things. And then we can go and calculate the genetic effect, what's heritable. And then because thanks to some really great scientists in our area, John Blangero's developed methods where you can correct for the genetics, and then all you got is the environmental effect. Now it's a matter of, okay, now we get that same participant, or population of participants, and we start using mass spectrometry to identify all the different types of exposures, right? And so when we do that, we can start looking at, think about 1,000 molecules. We can see that all of those molecules, let's call it exogenous molecules, what their phenotypic association is to any phenotype we want to look at, right? And we have models and ways of integrating or kind of looking at that correlation, right? Those associations, right? And then you can take that and then kind of put that into other gene bioenvironment interaction models, and then calculate that as well, right? So there's definitely an approach which requires a lot of really cool statistical heavy statistics to calculate this, find these associations, and then see how the genome interacts with the environment. Yeah, but it makes you look at all of the contributing factors to these diseases. And the coolest thing that I've seen is you can actually see all these molecules and make these associations to either known biomarkers, right? Or the molecules and their association to, for example, a disease like Alzheimer's, right? And they can do calculations where you can quantify the effect, right? So what is a causal effect, right? What is driving these phenotypes, right? But what is also protective, right? What's in the kind of negative domain, kind of to simplify it, right? What's positive and correlated, but what's also negatively correlated? Because then that's a protective response, a protective factor, right? So you really can pinpoint down to what these things are doing in a larger population, in an individual variation, and then also to the larger population, right? To see how it kind of stacks up, right? So I'll tell you what, I don't think I could ever go back to regular basic science ever again, because if you want to talk about precision medicine and translation, this is it. Yeah. So it's very, very, very fascinating. Very, very direct to the community, because now you can disseminate, you're studying the population in the community. Hey, guess what? Watch out for this stuff. it's directly, actionable policies can be used from this type of data. Yeah. Yeah.
Yeah, definitely. It's nice. Like, I mean, the idea of having the family studies is very interesting, because what you're doing is basically you're reducing the noise, like the genetic noise that you would have in a huge cohort. You're basically simplifying the genetic aspect so you can identify
Yes.
the effects of
Amplify. Yeah.
the stuff, which is like the molecules of the environment. And
Yeah.
that's pretty cool, because it means that if you identify a molecule, you can, so in theory, you could create something to counteract the effect of that bad exposition, like in theory. I mean, I don't know if this is something that you're thinking about, or is it just at the moment it's more like I'm thinking of something like antibodies, for example. For
Okay.
some part of the exposome could probably be targeted with antibodies that you would inject, and that would neutralize, for example, what's circulating in your blood. That should be there.
Yeah, I mean, there could be, if you were able to capture, for example, let's say you looked at proteins, or if you, or maybe,
Yeah. I'm thinking more big stuff, not like small molecules at the moment, like more big stuff, like
yeah,
something that could come from, I don't know, from food, or something that could come from mushroom yeast, or something like a protein, for example, or a peptide that shouldn't be there. And
I mean, I would,
causing,
yeah,
yeah,
yes, if you found the molecule that was either maybe had a particular mechanism where an antibody could be a therapeutic, yes.
yeah,
Yes, you could. Yeah, you could. If you saw how it participated in that pathway, and maybe it was binding to a receptor like too much, we
yes.
found that, right? Overactivation in that capacity. Yeah, there you go. Yeah, but yeah, no, you're exactly right. Yeah, that
Yeah.
definitely could do that.
That's what I was thinking about. Yeah, that's what I work on. So like, there's a slight bias.
Yeah, no, but one thing that I've seen that is those protective effects. I mean, if you think about it, maybe these biomarkers, that when you identify them, because we're always looking like, oh, what is like upregulated in these responses, right? Or early indicators. That's another thing. But what about the things that are like down, right? They're down regulated, right? Those protective factors that, like, for example, let's say in Alzheimer's, you have a certain lipid panel, certain lipids or molecules, but so you can actually, if you think about it, maybe these lipids are protective through some mechanism. Maybe they're sequestering reactive oxidative species, right? But we could see that in these type of experiments. And then guess what? All that requirement is now take a supplement, right? Some what is it called? Like prophylactic, right? You could start taking preventative measures, and maybe it doesn't have to be a drug. Maybe it is some, uh,
Yeah, or the
type
kind of
of
intervention.
yeah,
Yeah.
like some type of like kind of mixture of lipids, or I mean, or fats, right? If you think lipids are fats, right? It could be that simple, right? Eat more butter. I don't know, you know,
Yeah.
get more cholesterol, because then that will help the conversion of various types of lipid molecules, lipid pathways. Yeah.
Do you have any results at the moment? Is there anything that has been published that, for example, links parts of the exposome to Alzheimer's diseases or diabetes or?
Oh, yeah. There's already, not us specifically. I want to say, I'm trying to think of some studies out there. Yeah, if you they're they're already looking at, so that's a good question. I'm trying to like think of, um so for example, like for these forever chemicals, these PFASs, right? Uh, they're in a lot of like flame retardants and like all kinds of, or I was gonna say plastics. Plastics are still, we know where we know they can be contributing, and they can kind of get it. They can cross the blood-brain barrier. They can end up in a lot of places. But probably there's already like, I would say, several molecules that if you're exposed to, there already have like direct association. Though probably the ones that are the most studied are the particulate matter. right? When we inhale, which there's a particle larger, they're kind of like, they can range from like 10 nanometers to 2.5 nanometer particles, right? If they come out in emissions, right? Those are probably the most studied. We know that those can have a direct link to a variety of diseases, right? There's already been a lot of association studies. Polyaromatic hydrocarbons, stuff from burning, you know, the mycotoxins have already been shown to be associated with liver disease and now liver cancer, but now also Alzheimer's. There's a lot of research in that now. They're able to cross the blood-brain barrier. So they're already doing a lot of these studies, and there's already some stuff where we know if you're exposed to it, it's going to be super detrimental. You know. Yeah, I mean smoking, right? Think about it.
Yeah, I mean, smoking is the big one. So smoking is the one everybody knows about, and
Yeah.
But I feel like most of the exposomics so far has been on cancer. Like, well, it wasn't even called exposomics, but it was mostly like cancer, like association studies.
Yeah. You're right.
Yeah.
Cancer was I'm sorry. Go ahead.
No, that's it.
Oh, yeah. I'm actually, so that's funny you said cancer because we are now looking at the components of environmental toxicants, right? Things that are man-made, they end up in the blood. And there's some level of, they're probably causing neuroinflammation. They're probably causing a lot of these things. Well, a lot of these chemicals themselves are carcinogenic, right?
Okay.
One example is bisphenol A. It's found in plastics. There's a list of compounds that a lot of dyes. There can be like from food additives that you put them in vitro. You put them on cells, and you're going to get a cancer phenotype. Yeah,
You could do that in vitro.
yeah. There's been already several studies. Yeah.
You can immortalize cells with plastic.
No. You can. No, what I'm saying is like these compounds can actually stimulate. Not only are they genotoxic, they could cause genome instability, but they could also stimulate the cell to start to proliferate or to divide or to like migrate or to become more invasive.
So they stimulate proliferation.
Yeah. They can either. I mean, if you think about like mimetic molecules that are mimetic of estrogens. Yeah. Estrogen mimetics. They can they'll activate like hormone responses. You get kind of all kinds of transcriptional responses as well, and the cells will start to start to turn over and start to divide rapidly. So a lot of these plastic molecules and a lot of these like they can bind to receptors. So it's something we're also looking at. The carcinogenome or carcinogene.
Yeah. But I'm curious about this because I'm how sure are we that they're really linked to cancer?
Because they're toxic.
I mean,
They've been studied.
but there's millions of molecules. How do we know it's these ones and not something that comes with it?
Because they studied
another molecule. Yeah.
Well, they may be synergistic effects because think about if you inhale smoke, there's 7,000 chemicals in smoke. 70 of them are carcinogenic. Right?
How do we know? That's my question.
We know
because we know which ones.
Because we've taken them and we've tested them in rats and they get cancer. Yeah, there's animal studies. Yeah.
Okay.
So right now, there's a couple of databases. I have a student working on a project where we have like 7,000 compounds that we've identified, right? And that are, maybe half of them are significant, or or maybe there's some subsets we're looking at. But like Woody, she's cross-validating a lot of these compounds in other databases. So what are these compounds? Like, where are they found? Kind of categorizing them. Where are they found? A lot of them are industrial. A lot of them are in food. And then what what is there any kind of toxicity information on them? Have they been previously tested individually? And we get a lot of hits for some of these compounds actually causing a lot of cancer, or have been tested. There's quite a bit that have already been shown that you're exposed to that can cause cancer. Yeah.
Like how far? Because we can probably rank them. It's like to know, like we can the by correlation.
Well, they've been directly tested and they can be cancer inducing, right? So, and they can be categorized based on their level of toxicity. They are called group 1 carcinogens. So, World Health, WHO will categorize some, I think there's 4 groups. So group 1, group 2, group 3, group 4. You may have to fact check me. I believe it's 4. Yeah. But for example, aflatoxin, which we're looking here in the Rio Grande Valley, is a group 1 carcinogen. Yeah.
And where does it come from?
Fungus.
Yeah. So it's not. It's not a chemical.
It is a chemical.
It's not a. It's not like an industrial chemical. It comes from.
It's not a man-made. Yeah. So that would be called an environmental toxin. Yeah. So. Yeah. But bisphenol A is a group 1 card. Benzene A pyruvate. Sorry, not pyruvate. Oh my God, I'm drawing a blank. Bisphenol A has been shown to cause cancer. Oh my gosh, I'm drawing a blank on a couple of the ones. We have some other scientists working on these. Benzoate. There you go. Carcinogenic.
But aflatoxin is very interesting because can you talk a little bit about aflatoxin? Like, just what is it?
So unlike the other ones that are kind of man-made, there are a couple of
because it comes from fungus.
There's a variety of fungi, and there's a variety of these molecules or mycotoxins they call them. But the ones we look at is aflatoxin, and there's several. Aflatoxin is also aflatoxin B1, B2, and it basically gets converted, or there are different variants, right? There's actually, if I'm not mistaken, there can be up to 23. No, maybe I think now it's 26 different metabolites or versions of this particular aflatoxin, right? And basically what happens, it's a secondary metabolite in the fungus itself. And so in Aspergillus, it exists in Aspergillus flavus and parasiticillus. However, let's say it grows on corn, rice, a lot of different products. It can be found in meat, milk. So what happens is when you consume it, aflatoxin, B1, it's inert. But your P450 enzymes will biotransform it and make there's a position on there, and it basically forms an 8,9 hypoxy, and that hypoxy site now becomes super reactive to now it becomes super toxic at that stage. Now it can undergo a series of mechanisms to where it could also be eliminated by your phase 2 detoxification mechanisms that the body has, or it can go down other pathways, most through your genetic kind of vulnerability of some sense, or the amount. That could also be a reason why. But then it can it can form other modifications that then allow it to actually bind to the DNA. Yeah. So then it becomes genotoxic. Now, guess where it binds to most often and favorably to? So guess the gene. P53.
Oh, P53.
The tumor suppressor.
Yeah.
So, right, and basically make it inactive. Guess what? Your tumor suppressor is gone. It's happening in the liver. Boom. Liver cancer. So, and there have been data to show that it can exacerbate high doses, So at some point the cell I was actually talking about this recently. at some point the cell, and for a lot of different toxins, is like, whether it's a slow, gradual, chronic exposure or like a super high dose, at some point the cell is like, I'm done. I give up, and it just cannot, handle this kind of like environmental load of toxicity. And so then it starts going through other pathways. It can activate reactive oxidative species. So you can have a whole cascade of just this one toxin beyond genotoxicity. Yeah. So.
Okay, and this has been shown.
Yeah. Oh yeah.
Yeah, the association between aflatoxin and liver cancer is something that's known.
Oh yeah. It's probably probably more well studied in Chinese populations. A lot of Latin American populations are looking at it. African populations, yeah, where either the genotoxic mechanism can it be more prevalent. They have seen it in the Hispanic population in South Texas, and also associated with liver cancer, but kind of less frequent. But they've also not studied it as much, right? The surveillance is pretty low. So that's what we're working on. That's what we think is happening here. But it also may also be acting synergistically with maybe the viruses here in the liver, right? So they've shown that certain types of viruses along with aflatoxin will have a synergistic effect, and you're just going to it's just more carcinogenic. And so it's all coming, it's the liver, if you think about it, is the filtering organ. Everything goes to the liver. And so once you start messing up that, then you're, game over, right? Yeah, but it's a perfect example of like an exposure, right? Where your body is actually making it to become more toxic.
Yeah, and it's the liver is probably the organ that's most affected by the exposomes toxicity, because everything goes to the liver. Everything filtered by the liver. Yeah,
I wouldn't run away from that blanket statement. Yeah, the liver. You need a good liver, probably a healthy body. Yeah.
Okay, that's good to know. So, but why did you choose to work on aflatoxin?
So several reasons. John Blanchero said, check it out. And he's always typically right about stuff. So there's evidence. So here's the kind of, if you look at like kind of signals out there, they don't know, there are higher rates of fatty liver and liver cancer in the Hispanic population, right? It just so happens that aflatoxin loves to grow on corn. And there are plenty of studies that show Hispanics, especially the Mexican Americans, eat like 3 to 4 times more corn than anybody else. So it's a beautiful hypothesis if you think about it, more aflatoxin, more corn that Hispanics eat, And like I said, there are some studies that have shown that it's been actually tested on crops here in the Rio Grande Valley. And then also, it's also been found in the Hispanic population. Yeah.
Okay. That's a pretty good example because if it's coming from the corn, then there's a strong correlation between the amount of corn people eat and being Hispanic. So you might put that on the genetic aspect, but it's actually not genetic. I'm simplifying, of course, but it's So you might give too much weight to some genes and say, okay, if you have these genes, then you're more likely to get fatty liver disease or liver cancer. But actually, it's not just the gene, or the gene might have like a very small effect, but it's more like what you eat and the fact that you eat a lot of corn and therefore you can have a higher concentration of alpha-toxin, and that higher concentration might be the most important explaining factor of the liver cancer or the fatty liver disease.
I think both can be true too, because maybe our genes. But no, that's right too.
True. But if we just study the genes, we'd never know.
100% correct. Absolutely.
Yeah. So we would put 100% of the weight on the genes.
Right.
Yeah, for explaining disease, and that might be wrong. And
There's an interesting theory with aflatoxin because it does that it seems that they've tested this in animals that there is this ability, and I kind of mentioned it earlier, where the cell can no longer, it basically reaches a capacity of being able to handle aflatoxin, to detoxify it. Yeah. And it could be cumulative, right? It's additive, right? And then you just keep.
yeah, it's another. It's
Yeah.
yeah,
And it doesn't
not
matter.
for life.
So, yeah. So at some point, you're just like, I'm done, right? And now that's when the problems start happening, right?
Yeah, and that's actually a very interesting topic, because it's if you look at ethnicity, for example, the ethnicity can come with some specific genetic signal, but it also comes with a specific cultural signal, more often than that genetic signal. And yeah, and deconvoluting the two is actually a hard problem. So what comes from the cultural habits, for example, how you eat. How you eat is a big one, because it's like cultures are very, very. I mean, everybody loves the food of their own culture usually, so it's like
I mean,
a very defining factor of cultures.
barbecue,
Yeah.
barbecue and charring meat.
Bar-
Oh man, yeah, smoke. Yeah, you're right. Absolutely. I
Yeah.
wouldn't. Yeah,
Yeah,
absolutely.
exactly. And when you take something like Hispanics have a huge variety of genetics of Hispanics is huge. It's like you have people from African descent, you have people from different parts of Europe, you have people, and they all consider Hispanics.
Right.
So yeah, so you have this huge diversity that just put into one box called Hispanics, and yeah.
I
So,
think what.
but maybe it might be yeah. So that's what I'm saying. Like, maybe if when you're measuring something, it might be something else. Maybe it might just be that they eat more corn, and it just so happens that in that region there's alpha toxin on the corn.
Right. Yeah.
Yeah.
Yeah. No. Absolutely. I think this is the way you probably should look at, disease and medicine, right? Kind of you see it from both ends, But yeah, no, absolutely.
Yeah, yeah. This is something I've been thinking about like the limitations of categorization of people into different boxes. Um, yeah.
Oh, right.
So
Yeah.
yeah, because I think it ultimately does a disservice to the research, because it's like too at a certain point it becomes like too broad a category, so it becomes like blurry. You have
Yeah.
people that are very different, so they still consider culture the same. So they culturally considered to be from the same ethnicity, but they have different genetic backgrounds.
Right.
And
I
yeah,
think
Africa is a good example.
well,
Africa is a perfect example. It's like the huge genetic diversity, and somehow they all end up in the same box.
no, yeah. I've seen a lot more Hispanic studies where they're kind of categorizing them as like, for example, Afro-Cuban,
Yeah.
Mexican American. But you're right, where you live, right? There's probably less. I mean, they just sequenced the Moroccan genome, right?
Part of it, yeah, part of it.
Part of it, yeah. So
Yeah.
it's only part, but yeah, we don't. The more genetic sequencing we do, all we're looking at. I mean, if you think about it, like ethnicity and all these things are kind of social constructs, but we're just looking at variation, yeah, and how everybody groups together, right? But
Exactly.
and the
Yeah,
part of
like,
and the environment is where it comes from too. So you know.
yeah.
It's people don't really think about it, but I think they are. And there's more people now trying to like study this. We're the early adopters, but I think it'll become more and more. I was actually talking to some of my, mass spectrometry friends. They're like, well, what the hell what's the exposome? Never heard of that. Yeah. I only found out a couple of years too myself.
Mhm.
But there's a journal of Exposomics. And there's been some really big breakthroughs within the last like 5 years. Um, Maybe people don't realize they're doing exposomic research, right? So, I mean, it's toxicology 101. Yeah.
Yeah, I mean, toxicology just became part of Exposomics. If it's you can be exposed to. I mean, toxicology is part of it.
Exactly.
Yeah, it's okay. I think so.
That paper we published, when the one at MCP, was the paper before it in 2021 with Nicholas was the first author. I was calling it the reactomics, the venom reactome, but that's the venom exposome. I didn't even know I was doing exposomics. Yeah. Yeah.
Yeah, it's right.
Yeah. So imagine when I come and I'm working now with this group at, ETRGV, and they're talking about the exposome. I'm like, oh my God, I just did that experiment. I'm doing that. We did this in mice. Great. So, but now it's like a larger scale. So,
Yeah, you could do the same thing. You can probably do exposomics working on accessory vesicles like EVs. They're probably a very good.
Which they've shown. There's a couple of people we're collaborating with a really amazing professor out in the University of New Mexico. But there are people looking at kind of acute exposure and the extracellular vesicles capturing that injury. And we can see with that's the extracellular vesicle before and after injury is completely different, whether it's the surface molecules, whether it's the microRNA inside. So not only is it it's a unique biomarker in terms of its composition, but it's my hypothesis, and there's some evidence that shows it's probably transmitting that signal to the extracellular vesicles with cell communication, right? So it's transmitting that toxic signal and making all these other cells sick too, right? It's spreading that pathology that had an origin, right? So,
or it might be part of the cure also. Might be part of the response.
Right?
Yeah. Yeah, it could be. But in any case, it would be a biomarker. So it would be something that you can use to identify the injury and the exposure. Yeah.
It's definitely biomarker potential, but then also gives the added mechanistic ability, features to then look at mechanism, right, as well. Sometimes we don't really think about it, but those biomarkers, like, what is their function? Why are they responding in this way? To explain that particular chronic disease or acute injury, right? Yeah.
I think that's one of the most fascinating aspects of fundamental biology, is like really finding these mechanisms behind that explain the phenotype.
Yeah. Yeah.
That's I think that's what fundamental biology is all about. That's what I like about it.
Yeah. Oh yeah.
Yeah. That's yeah. So if I think about all that, there's the broad population study, then there's you can separate them by ethnicity. There's the family study that's even more precise, and so there's this funnel. But I think we should like statistically. I mean, it's hard. I'm not saying it's easy, but I think we should start thinking more in terms of genes independently and then together, instead of thinking of boxes of people. I think that would be much more informative. This is, I mean, we start to do that because now we have tools to start doing things like that. Like with CRISPR screens, we're no longer on the single knockout.
Right.
So yeah, so that started. But I think we should start thinking more in terms of like not people, but building blocks of responses.
Right. Yeah. And also maybe not even categorizing
Exactly. Yeah.
diseases. And I think this is, and I know you talked about this, like normal aging versus like Alzheimer's, right? How do you, because this gets complicated and it gets noisy. Like, for example, mild cognitive impairment, right? Or behavior, or for example, like, people who are for drug addiction. How do you reduce the biochem? How do you reduce that human variation and noise, and just to capture the signal, right? But you don't want to really categorize because you're biasing for something already, right? Maybe it comes down to some knowing the genetics and looking just purely at the exposure and then making these correlations over just a larger subset of individuals, right? And then instead of like, I guess, clinically classifying them, there's maybe a biomarker of a known, something we can, it's almost, we think about it, reducing it to, A plus B equals C, but across thousands of individuals, right?
Across thousands of individuals and 20,000 genes and and a few million molecules that you can be exposed to.
Yeah.
Yeah.
Yeah, no, it's complex, man. It's complex, but that's why AI and is gonna be super important for this.
Yeah, such
Creating models. Yeah.
we can't.
Well,
We have to talk about it.
a lot of computing power. Yeah. It's gonna be essential for this.
Yes, I mean, I think now we can do it. I think now we can do it.
Yeah.
There's and it's because our computing power is orders and orders of magnitude bigger than what we had even like 5 years ago.
Yeah.
It's not even on the same scale.
Right.
And, yeah, so it's interesting because it's really like the defining factor. When you look at the algorithms, they haven't changed that much. It's like it's still mostly the same theory, the same thing that we're using. However, we have big machines, like we have huge machines.
Yeah.
And we can definitely run to like millions of molecules like pretty easily through that. The challenge, in my opinion, seems to be that comes from my experience because I was trained. My original training is as a computer scientist, specializing in AI, math, and then in AI.
Yeah.
And so I made the jump to start working on biology, like which is like exclusively what I do now. I only work on biology data sets and things like that. And when I started, I had this this illusion that basically all data is the same. You have the algorithm. If you have if an algorithm works on images, it's also going to work on molecules.
Right.
And it's absolutely not true. It's completely not true.
Right. Yeah.
I know that by experience, like it has been shown every time for everything I've done, it's different. You need that domain knowledge. And I think this is really what's capping us when it comes to AI. It's like a lot of what we do when we apply it to biology or chemistry is basically we take what we do on NLP, natural language processing, or what we do on imaging, and we try to apply it as is, and that with slight variations. And that's not necessarily the right approach. And I think that's the big cap because we definitely have the computing power. We need to develop the algorithms to make them more able to handle that type of data. And it might be like big changes, or it might be like small tweaks. Like AlphaFold is, for example, a small tweaks on transformers.
Yeah. Okay.
So, it's basically a big transformer, but that uses the orthology, like the fact that you find similar proteins across species.
Yeah.
You take that basically as your database and you train a transformer. Like I'm simplifying, but that's basically the idea. And that's the tweak that made AlphaFold like the first version possible.
Right.
So it might be something like that. Just thinking about it differently.
Yeah.
Like knowing, like, because when you think about it, it's interesting because it's like one piece of knowledge from biology. Like you can have and it's something that every biologist knows. And it's like you have a protein that's in humans, like a gene. That gene protein is like basically the same thing. Because genes goes like this. Yeah, genes make protein. So you have that gene, that protein inside in the humans, but you also have the equivalent in mice. And you also have the equivalent in rats. And this is why we have mice models. And you might have the structure for the mouse, but you don't have the structure for the humans, or vice versa. Basically. So and they're very similar. They're not the same because it's different species, but they're very, very similar because they have the same function. So the idea is pretty simple. I mean, why not learn from what we have to apply to what we don't have, because what we don't have is very similar to what we have.
Right.
So that's basically the idea of AlphaFold. That this is something that is like it's basic knowledge in biology, but that's not necessarily basic knowledge in computer science.
Right. I guess.
Because you never get to work on proteins. You never get to work about autologous proteins. Maybe think about that. It's not part of what you do. So maybe we need this small tweaks, like just like micro tweaks, like small tweaks here and there, and then we can probably handle the exposomics.
Yeah. I mean, I think in anything, because you develop methods, right, for any kind of science, and maybe it's not, but you have another biological question, and you try to apply methods to that biological questions, but maybe that's not the best way. Maybe like, kind of first principles thinking to the biological question is better, to develop original methods to target this. That's fun. That's kind of fundamental, but I sometimes I know we don't think in that way, whether it's cost or speed or whatever, but I used to develop methods back in the day looking at for proteomics and mass spectrometry. Used to hate it, but now it was probably the best training I ever got, because it was like, because it doesn't allow you to go further into the biology, but you're able to first capture that biological feature that nobody else could, right?
Yeah.
I think we need more. You know what? I think we need more people doing that. It takes more time though, and I think we need to have more devotion and dedication to doing that, to really get to these measurements and these methods that are, it's not a they're going to be specific, and they're going to be more accurate in trying to defining some of these biological mechanisms. And I think that's also similar to, adjusting these models, these computational models or statistical models for biology too, right?
Yeah. Yes. I'm very big on methods.
Yeah. So I think it's just, honestly, I think it's just going to get better and better. There definitely is going to have to be rigor, and not using data sets that have a lot of noise. Yeah, I mean, you're familiar with this, batch effects, reducing that, even the way samples are collected or data is collected, the bias and variance in that alone, right? But I think it's getting way better. I am so jealous of my students and how amazing the quality of the chemical measurements that they do. I mean, I was like, oh, I cannot believe it. It's their, mass specs. Even now, still, it's like throwing a rocket at an airplane and just go through crashes. These instruments are so finely tuned. They're so delicate, but still, and you look at them, they're hardy, but man, it just takes one fluctuation in temperature, one fluctuation, in a vibration of the building, and it's like your mean goes, it's like, and you're game over. Yeah. That's why I get annoyed when people call them machines. They're instruments, right? They're super precise, but they're, like I said, it's like throwing a rock at an airplane and it crashes.
Yeah. It's
But
same thing for AI. It's I think like methods and model development, I think, is the way to go. And what's super interesting is that you end up basically like you use biology. Everything we do on the lab is like new models that we basically embed, because biology gives you these interesting problems that force you to innovate.
yes.
Yeah. And that's really cool. Yeah.
So one thing we're doing, and that's a great segue into this, because one thing that we're doing, so we are implementing mass spectrometry tools and methods in large scale population studies, right? And I told you I have worked on method development. It was for more cellular approaches at a time when we couldn't even really do quantitative proteomics. We couldn't do it that great. And now what we're working on in terms of, I would say, kind of method and biological development is actually looking at different biological sources that can actually be used to capture the true I don't want to say true, but specific biomarkers, right? Less invasive, less, more direct measurements of exposure, right? So for example, like hair, you know, um, you know, using hair to capture, it's a metabolic process, but you can actually use hair and capture like kind of the early or kind of uh, kind of like short-term exposure, and also you can grind up the hair and capture what's been exposed to you long-term, right? Because it is a metabolic process, right? They've already been doing it for illegal drugs, but you can capture a lot of other compounds in there too. So but we're able to do that through with mass spectrometry at like super high speeds. We capture a diverse set of molecules, right? So developing these methods that can then be used, because they can then be used like in a larger population. And maybe we're going to capture, more relevant biomarkers. One thing that hasn't been looked at is like, for example, mothers and breastfeeding, right? The biological question is, what is that early exposure and development, right? Women who, maybe women, a lot of women are on antidepressants. Maybe they're in other parts of the world where they're getting lead poisoning, right? But they're transmitting that through their breast milk, right? So that's a study we're actually getting ready to start. So I think it's going to be a combination of like these unique the biological question where we're getting the answer, or where we're trying to like probe into, and then a more highly specific method, chemical plus mass spectrometer based method. And maybe not. Maybe it's kind of ELISA or Western blot or something like that, an immunological type assay. But you get to the better, the better readout of, yeah.
Yeah. I don't think you're ready for the ELISA.
Yeah, I know. There's some really great instruments that can do like high throughput ELISA. We have actually a couple that can do whole panels. So Quantarix is or maybe, you know what? Or maybe we don't even do mass spec. We can do, Illumina has this new proteomics kind of platform. It's a no,
You don't. If you don't mass spec.
I'll do another type. Well, you can still do proteomics. It'll just be a kind of aptamer or antibody based method where you're capturing all these metabolites, right? With a different kind of platform. Whatever, it doesn't matter, honestly, to me, as long as we capture that real, biological response, right?
But
yeah,
what would be the best method to capture, for example, like breast milk is it strikes me like a super good idea, like an amazing idea, because obviously you don't want, there's the whole transmission from the mother to the baby, but there's also like the fact that it's very easy to get, breast milk. It's like so what would be the best method to get like the most precise panel of exposomics that you could get, that would get from breast milk?
It's a great question. I don't think we're there yet. What I've seen kind of just from there would be a couple of, I don't think there is a best method yet, or maybe there never will be, because of the chemical diversity of what you're trying to capture. For example, just changing the solvents can get a whole nother set of different molecules. Just going from GC to LC, a whole nother panel of molecules, right? Even the computational libraries we use that can influence what we find, right?
Yeah.
What I think will happen and what we'll do is we're going to get faster and cheaper to then just do a multi-analyte analysis with a quick turnaround with more definitive markers being found. Yeah. Just to let you know that when we look at these small molecules, of the billions that exist, we could only capture about 1.4, about, what do they say, 1 to 2%. Yeah. There's so many things that we cannot see with a mass spectrometer. or they can be detected, but they cannot be defined.
So you don't know what
We don't know what it is.
Yeah. So, but what does it look like? Does it look like a random? It does have. Yeah, I mean, it can be random because
You want to know? We call them ghosts. We call them ghosts in the lab because it has a mass, it has a retention time, it has all these properties, and it's blank.
what's the issue? Is it because it's not in the database?
Yes, it's not in the database. It has never been defined yet, and it hasn't been validated in a lot of ways. Yeah.
Okay. Seems like a nice project to work on.
Take a look at it, man. I'll send you. Yeah. They're developing some tools now to where you can get these, like, you can kind of have predictions based on all of this data we can collect, and kind of better have kind of create your own identification scheme, I guess. There are people who are working on this. Yeah. Yeah, but it is a severe problem. But not a lot of people are working on this, so it's kind of slow going now. There's only a few exposomic centers that are, in the world, well, in the United States, but there's only a few groups really tackling this. Yeah. So, but yeah, that's a huge limitation. Huge limitation. Yeah.
Because I don't see why. I mean, if you can see the signal, if you get the signal from the mass spec, there must be a way to identify what that signal is.
No.
I mean, there must be one. I mean, there aren't it yet. There must be one. Wait, there must be a way. There must be a computational way to do it. Yeah, I think. I think.
Well, so one of the issues is, and there's several. One of the issues is, for example, you can have the same mass, same retention time, a lot of the same properties, but the structure is different. So how do you know which structure, right? Because structure can also be a different. Yeah, so the structure can also be a different molecule, right? It's got the same chemical composition, same mass.
And they also, yeah. You don't know the orientation also. You don't know the orientation of the chemical.
100%. Yep. You don't. Yep. When you take drugs, when you take a pure drug, you get that's even in itself, a racemic mixture. Yeah. It's not even pure either.
But,
yeah.
But if you get the sequence, that's
a
good start.
That's a peptide. Peptides are a lot easier. Yeah.
Yeah. So okay.
So metabolomics.
Let's say
small molecule.
Okay. So peptides, if it's small, like it's basically linear. So you know basically the structure.
Well, no, the amino acids in themselves have a unique mass, right? Except for leucine and isoleucine.
Yeah. But if you can identify different amino acids, if the peptide is a short peptide, it doesn't have a complex, like a 3D structure by itself.
No. Oh, no.
Yeah.
But we do MS2 fragmentation to know what it is. Yeah. We fragment it again into smaller parts, and then we can kind of rebuild that sequence. But those amino acids have a unique sequence, right? And we're not fragmenting the amino acids themselves. We're fragmenting them in sections. So we can get that. Those masses are so unique to this sequence. Yeah. So it's easy to define it.
But if we're talking about molecules, like small molecules,
right,
that would be a challenge. Because peptides are pretty big. So
small molecules are the challenge. Yeah, because for example, let's say you have, I'm trying to remember this compound. I actually teach it in my course. Like, if the hydrogen is coming out of you, or it's a methyl group, if it's coming out of you, right? It's one molecule, but if it's coming away from you, it's a different type. It's pretty much like a hallucinogenic or something. That happens a lot. Yeah. I'm kind of blanking on the molecule. It's kind of anyway. But that's the kind of, like, where the structure and the orientation, the kind of it's kind of like, orientation, right? Stereochemistry, is that correct? We're gonna have to fact check that. I believe I have a chemist here hanging out with me. how these molecules are on the, on the plane. If they're in or out, completely different molecule, completely different activity. Yeah. So same chemical mass, same chemical formula.
Yeah, you wouldn't be able to see the difference on the mass spec.
Yeah.
One would fit. Yeah.
Yeah.
The one would fit on a receptor and the other one would not.
Right.
Yeah.
Yeah. So we're still kind of in that space. There could be differences in fragmentation patterns, maybe. Or maybe there's a condition where you could fragment it and they'd be different. But that takes like more work, more adding to the database to see what that is. And then guess what? Those databases are not all the same across platforms. It's very like, not, it's still not a kind of unified method. There's no unified method for a lot of this stuff. That's the wild west.
Probably. Yeah, but there's probably a way. Like, I mean, stereochemistry is like, it's a hard problem. I don't think you can see that because you're basically you're measuring the mass over the charge, and the mass over the charge of the molecules like this or like this is basically the same.
Mm-hmm. Yeah.
So
you can't tell.
So you wouldn't get. Yeah, you can tell. You can tell when you get the orientation. But however, I'm pretty sure you can use the existing databases to be able to identify, like to give you at least an idea of what's the composition of the molecule. Doesn't have to be a ghost.
Well, but if there's some level of uncertainty, it won't define it. It'll fall out.
I know, but that's the current state of the art of the machine. But I think we should work on that. We should.
I can send you a lot of ghost molecules.
Yeah, send me some ghost molecules because I don't see any reason why they shouldn't exist.
Absolutely.
Yeah, we could probably like figure out an algorithm to give you an educated guess about what the ghost is.
Yeah. Okay, let's work on it. That's cool. Oh, you'd be super famous, and probably if you patented that, new technology.
I was thinking about the paper, but why not?
Yeah, I mean that would be a great and going back to what I was saying, maybe to find the ghost is to know the reference genome to those molecules or to those masses, or account for them at least, and maybe something could come up. Or if you found this molecule with a variety of other molecules, maybe that could infer what it is too.
Yeah.
I'm just talking out of my ass, but
that's a good use case for AI.
And 100%.
Yeah, it's a super good use case for AI, and I think it's a stepping stone. So what were we talking about like earlier, like feeding all these molecules and for the exposomics, but it doesn't work if we can't identify them. So first we need to identify them.
Yeah.
And so I think that would be like a pretty good use case for AI, like improving the identification, no more ghosts.
Right.
At least educated guess about the ghosts, and then we can get an even better data set for the exposomics. So I kind of see it like in two steps.
Oh, bro, let's do that. You would refine the data with that, with that method. Yeah, for 100%. Yeah. Yeah, absolutely. Yeah. Yeah, we should talk more on this. No, that's definitely, it's a global problem in metabolomics research. Yeah.
I think that's the number one problem is that because now we basically talking about something like you said, like gene overexpression, like gene time expression. It's like the space is huge.
Yeah.
If you had like the dimensionality, like to use a mathematical term, like it's huge, it's like immense. So it means that we need data.
Yeah.
The bigger the space, the more data we need.
Right.
So we can identify basically. So because it's always the same thing. Like this is something that's true all over the place when it comes to AI and math in general. It's like playing chess, basically, or like images. It's like the same thing. It's like everything is the same to a certain degree. That you have, if you imagine an image, and images have pixels.
Yeah.
And there's an infinite number of images you can generate. You just like with random pixels. But actually, like the images we see, they're not random. It's just a subset of the totality of images you could generate. And that's basically what usually called the manifold. So the game is basically to find the manifold of what's plausible and possible inside that huge space. Yeah. For that, we need data.
Yeah. We got it. We have it.
Okay, let's do that. Let's do that. Let's find a way to improve the identification of this. I was really step.
Yeah, that'll be a big impact. That's like, yeah, huge. Yeah. I'll send you some stuff. I'll send you people kind of some of the kind of like big questions in Exposome, like but Metabolomics. I mean, it's a huge problem, huge limitation. Yeah, we get about like almost 10,000 compounds, but we can only identify like 4,000. Yeah.
Okay.
What's the other stuff? Yeah.
Yeah. So what do you see as the future of Exposomics?
Exposomics on a chip. I see.
On a chip, so you're like moving around with something like.
Yeah, biomonitoring.
Yeah.
Yeah. I think
like an instrument that you move around. It's not just like your mass spec in your lab that's huge, but you have like a small thing that you can.
Or you're wearing some device that you can then send off to get read, and you can see what your kind of exposure is, maybe.
Okay.
Or when I say exposomics on a chip, I mean something where, for example, let's say you can have a quick reference to, like, as an assay to see, like, maybe common exposures, right? Could be unique in that. Microfluidics being integrated into this, right? Better knowledge, an exposomic score that can be read, maybe reference readings to where, like, if you know you're exposed, but maybe this is like baseline or not significant, right? It's like background versus like, what the hell? Like, this is bad. I don't know, maybe you're working in some place, and yeah, I could think a lot of this for occupational like health, and maybe people in various industries, they have to biomonitor, radiation. I mean, think about radiation exposure.
Yeah, so the same thing.
Basically, yeah, health seems to have content.
Other than Geiger, Exposomics.
Yeah. But now we're looking at it in terms of low dose, chronic, and then constant, right? And monitoring in your house. We used to say that what's coming through your air condition.
But that's it's true. But that's slow. It's like that's the thing. Is like it's like a small exposure which probably do nothing, but over a long period of time it can be very detrimental. So it's I mean, if you have a like this kind of device that tells you, okay, this is probably a place you shouldn't be in, but you might be just fine, like being there for like depending on the type of exposure, it might be 10 minutes, might be 2 years.
Yeah.
But so yeah. So how like the temporal aspect of exposomics makes it very tricky to deal with. Because it's a build up.
I mean,
or is it? I mean, yeah.
Not necessarily. I mean, not necessarily, because, like, for example, like so just being exposed to, so for example, like heavy metal, like you don't want to be exposed to methyl mercury, you'll die. Right? Get it in fish, right? So it's not. No, you can get acute exposures that can just take you down like super quick, or stuff that causes cancer pretty fast, right?
But,
but even arsenic, lead, lead poisoning.
These also are usually pretty easy to identify because the person dies or get like sick very quickly.
Yeah.
So it's the long-term effects that are actually very, very hard to find.
Yeah, right, absolutely. But I think we have enough data now, and I think we'll be able to see or even predict based on the structure of some of these, like, okay, stay away, don't, right,
Yeah.
I know that there are estrogen mimetics in certain types of beers. Don't drink those beers because I do not want estrogen in my.
It's like phytoestrogens.
Yeah. So.
Soy also has that.
Yeah, I know. I don't need soy.
Plastics also.
Yeah, processed
Yeah.
foods. Yeah.
Yeah. I mean, that might be one of the reasons why we see a decreasing, like testosterone is decreasing across time. That might be it.
I
Part
will.
of it. I don't know.
I think looking at the exposome and trying to mitigate or maybe even promoting bioremediation or
Yeah.
or strategies individually or at the population level has commercialization value, but also like implications for health and disease too. So
Yes, the testosterone aspect is
I mean
very
that
interesting. That's
yeah.
that's a whole question in itself. It's like and not a lot of people are working on it. It seems. Just
They no, there's a lot of people working on
there's
it.
a lot of people.
That's another podcast for yeah, it's another
I know it's another.
we don't study it. We don't look at testosterone levels.
Yeah.
But there's a lot of evidence showing that it's historically.
Oh yeah, because there's evidence showing that it's dropping, but why is it dropping? That's what I'm. Yeah, it's like I don't see a lot of maybe these papers exist and I don't read them.
Yeah.
But I honestly don't look. but 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 it seems to me that it's there's not that much research.
There's a couple of good the phthalates, the plastics can have feminizing. They're endocrine disruptors.
Yeah, I've seen these ones. Yeah, I've seen these ones.
So right there,
Manufacturers.
that's a great place to start.
Yeah, but it seems to be more complex than that. And Exposomics might. I mean, it seems like a perfect use case for me. I mean,
Oh.
it's also like it. It's also very complex
You
because
could.
you have it's decreasing across time. So I don't know if you have a data set of Exposomics across time, or if I do this, it gets thumbs up.
I don't know that. Well, all you would have to do is to look at the individual, record their testosterone, and see what was in their exposome to identify the correlations of the molecules in that.
Exactly, and we need to, but for that we need to have like we need to to record testosterone systematically.
Yeah.
Yeah, and this is not something that we usually think about. Like in
I do when I go to the
studies.
doctor.
Yeah, when you go to the doctor, but when we design a study,
Yeah.
we
Yeah,
don't
no,
do that all the time. Yeah.
no, that's a good question. I wonder if they're, I think it's, man, we should definitely look into that, the exposome and testosterone levels.
We
Yeah.
could, we could do like all sexual hormones.
Yeah. Yeah. There's
Yeah,
sure.
because we-
I know there's some evidence, but yeah, no, I've never seen, I haven't actually looked at a study on that.
Yeah, yeah, and it's interesting also because you like just anecdotally like talking to people and men telling me like they've been to the doctor and they're usually like over 40 and they have a low testosterone level, like even like some younger people, but you wouldn't tell just by looking at them.
Right.
Also interesting, yeah.
So one of the things too, and I was talking about like these environmental domains, like stress, warfare. I knew some people. I knew people back in Boston who were at Brigham and Women's that were looking at veterans, and their exposure to these large guns and mortar fire was giving them I mean, getting hit in the head can actually can cause you to have low testosterone levels. So, and I know a lot of veterans coming back have lower testosterone levels.
That's interesting.
Yeah.
That's interesting. So if you get better up a lot, you use your testosterone.
Hits to the head. Yeah. Can have an effect. Yeah.
Wow, that's it's giving me theories about what might be happening in
Yeah,
these
I mean-
societies. You get bitten on the head, you lose your testosterone, you go down the hierarchy.
Man. Yeah. I mean, there's a lot. I need to take a look at. It's
Yeah.
really interesting.
Oh,
You know, causal effects for decreased testosterone, aside from just decreasing over time. I mean, it's pretty much peaks at about 18, 20, right? Sexual maturity, but then like I guess, I mean, also if you abuse steroids, you're not going to make endogenous testosterone, right?
yeah, that's
A lot of people are doing that.
Yeah.
So yeah, no, it's a fascinating area of research.
Yeah, if you abuse them, I mean that's no. Like if you take too many steroids, too much of them, you stop producing.
Yeah. I mean, maybe going back to the breast milk, being exposure early on
Yeah.
can have,
Yeah.
epigenetic changes in development.
Yes, because the steroids wouldn't explain it on the population level. On an individual level, I might explain the drop of testosterone, but like the whole population, not everybody's taking steroids. So,
Right, right, yeah. Yeah, yeah. That's, yeah, probably multi-causal effects. Yeah.
yeah, very multi-causal effect, probably, and this is interesting because it's probably something that has to do with chemicals, but not just chemicals. Probably like what we were talking about at the beginning. It's like exposomics, like the emotional exposome. It's like your exposure to yeah, could be trauma, could be anxiety, could
Yeah.
be war, could be I don't know, depression. All that probably has a huge effect on testosterone also.
Right. Yeah. Yeah. I mean, going to the emotional side, looking at, for example, like depression, um, I guess psychosocial disorders. Um, our group is looking at like how the pollutants and metals can affect, depression. Yeah. 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 They're showing there's already some studies that are showing this. Yeah. So once again, the exposome affecting your behavior, you know, yeah. Your mood. Yeah, for sure. Absolutely.
Significantly, so you can shows an effect on depression, anxiety.
Oh yeah. 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 Pollution. That alone, I think, is probably driving a lot of things.
Yeah, but is it really pollution, or is it because pollution comes also with a certain environment, like a more urban environment? Maybe you're living close
Well,
to a factory,
I,
and that's depressing. That could be depressing in itself.
yeah.
It's like,
Yeah. Wiki
yeah.
first, right? Yeah. No. No. Yeah. I hear what you're saying. Um. Yeah. It's hard now. These days, it's hard to define. Like, well, what's polluted, right? Is our what is allowed now? Um. Could maybe be considered, for example, the food we eat here in the United States, it's like Europe has nothing to do with it, right? The way they process food. Yeah. So we have food in a way, right? It's kind of how do you define pollution? What are the like set limits, the thresholds?
Yeah, yeah.
So
The U.S. food thing is fascinating because I don't think anybody knows.
yeah,
You see the effects, but I don't think anybody knows what's.
I don't know. I don't think anybody knows either. And well, I know we shouldn't eat them. That's probably one thing. And maybe this is an explanation of why we're seeing the United States kind of fall in these, on hard times, massive obesity in the United States, right? Close to 50% now, I think. Yeah, maybe it's a lot of people. A lot of people. When I was at American Society of Human Genetics meetings, we're looking at food exposure,
I
diet.
think food is the big one.
Beyond calories, beyond macromolecules, right? Carbohydrates, lipids, and like know what is in the food, right? Yeah.
Yeah, I think food is the big one. I think food
This is
is
the
like
yeah, this is the big thing on RFK, with his Maha studies and stuff. Yeah.
yeah.
Um,
Yeah, I think food is and it's also easy to measure because it's easy to take samples. You know what samples to take.
oh yeah. Oh
If you
yeah.
go into the environment, it's like what do you take? It's like you take a sample of the air, you take the water,
Yeah.
you take the soil,
Test
it
it.
becomes it becomes complex. But food is easy. It's like okay, this is food. Like take a sample and then
Yeah.
analyze it in the lab and see what's in it.
I could do that. I could actually just start doing that. Testing my own food.
It's like running that to the mouse like
You can do that. Yeah, it's not an impossible it's a chemical extraction. You can do it. Yeah.
yeah. So you digest and you. But if it's a peptide, then like how do you identify a peptide in a steak? It's like full of peptides already.
Say what? What do you mean?
How do you identify like a specific peptide? How do you know? I mean, you have a piece of meat, and we want to identify like
Because they already sequenced the bovine genome and proteome. They already have a reference proteome.
Yeah, but I mean, the reference is as good as the reference. It's yeah.
No, the genetic variance is already accounted for to make a particular protein. But if you're talking about small molecules, it would be no. It could be. It's a different. That's a different method. That's not protein. That's small molecules.
In theory, I think you still need the for the mass spec to work really well. You still need the exact sequence of what you're trying to sequence.
Oh yes, you do.
Yes.
Um.
So if you
Yeah.
because no cow is the reference cow. I mean, there's always a
Yeah, man, I don't know. Actually, I've only seen is it Boss Taurus? But I bet you there's a lot of variants that haven't been accounted for, right?
probably.
Yeah.
There's a huge diversity in cows. There's
There
is.
There is.
True. There's
Yeah. There's like the long-haired like cows up in the Scottish Highlands in England that have like super hairy. Yeah, they're not all. No, you're right.
small cows, fluffy cows,
The only thing I can say
cows.
Yeah. The only thing now is for mass spec in terms of proteomics, you don't have to use actually a reference proteome anymore. You can actually do it similar to metabolomics, and you can create a peptide library. So we can get one of those unique cows, chop it up, the meat, sequence it, or not sequence it, run it through a mass spec, and then the mass spec can identify what those sequences are independently. And then we can start, then we have, that's basically our library of peptides from an individual. Everybody's unique in that sense. Yeah. And so that's what you match up any kind of existing sample to this reference library of existing peptides.
Yes.
So
but how would you go about, for example, identifying like difference between Europe and the US in food? It's like it's
it would have to be.
It's not easy.
Oh no, that'd be hard. Yeah, no.
That's not an easy task.
No, because even within itself, you would have to like get a kind of like representation, right? Yeah, that's not easy.
And Europe is very diverse, like different cultures. Even in the same country, you have different cultures.
The way, yeah. That would be really hard to do. Yeah.
Maybe you could do, I don't know. You could do the whole of Europe versus South Texas.
Okay.
I mean, because if you take like the whole of Europe, becomes like a background, and then you take one part of the US, so you get a stronger signal. I'm just like imagining like that, and then you can identify something, and then you can do like other studies to see if you identify the same things in other regions of the US. Maybe. I don't know, but it strikes me like as a very hard problem.
Under the same at the same time at the same date.
I mean, that's also
That's also
I mean, you don't want to do it. Yeah, of course. Yeah, like if you do it on Thanksgiving, like everybody's eating very specific things on Thanksgiving.
Yeah.
So it's not very representative of and people eat the same things during winter time and during the summers. Very different.
Yes. Yeah, I think if you did it across crops, it would be easier. Crops, single crops, monocrops. Yeah.
So, yeah, but because it could be the crops, or it could be the processing.
Exactly. Yeah. No, you're right. But then you kind of define you can control more controls, right? You can look at several things, but but you'd have to break it down to a simplistic, comparison.
Anyway, this is a fascinating topic.
Yeah, thanks for having me on. I think people are gonna hear more about the exposome, what the limitations are, what the possibilities are. The conversation with you, other things to kind of work towards. Yep, I think we're just now scratching the surface.
Yeah. Yes, and I think the idea that we had about how to improve the mass spec identification, I think that would be a major stepping stone for Exposomics. If we can identify all these ghost compounds, that would be something. Yeah.
Yeah, it's a good title to the paper already, or the perspective, or however you want to like start it. Like, yeah, finding the ghosts.
Finding the ghosts. No more ghosts.
Yeah.
Yeah. Yeah. So
I'll send you those papers too, so you can take.
Oh, yeah.
Yeah.
yeah, send me the papers. Next time we can even go through the papers if we do get like a second episode. We could go
Oh
through the
yeah, yeah.
papers. Yeah.
5 episodes, 6
Oh,
episodes.
I mean,
yeah,
No, it's about research. Research is ongoing, so
exactly.
it's kind of slow. It takes time to get a paper out, but it's never ending. So
Right.
let's do another one. Let's do another one when we have something to say.
Sounds good, man. Appreciate it.
Thanks, man. Thanks for coming.
Absolutely, sir. You have a great Patty. Great morning. Take care, buddy.
Have a great night.